Hey there, Mr. P here. Last lecture we talked a little bit about redox reactions and what it means for a molecule to be reduced and what it means for a molecule to be oxidized. And now we're going to talk about why it's important to understand redox reactions so that we can then kind of build our understanding of these redox reactions and how they impact both cell respiration and photosynthesis. This is Chapter 8, Lecture 2. And we're going to talk about ATP production, specifically chemiosmosis and substrate-level phosphorylation. And the first thing that I want to talk about before we get into the specifics of chemiosmosis and the specifics of substrate-level phosphorylation, I want to talk a little bit about the electron transport chain. This molecule, or this series of proteins right here, would be the electron transport chain. And we call electron transport chain a bunch of electron carriers because that's essentially what they're doing. They're all proteins and they're all spanning the membrane. And so if I kind of drew a membrane through here, all of these proteins would be within the membrane itself, and specifically either the thylakoid membrane or the cristae membrane. Remember, the thylakoid membrane would be the thylakoids within a chloroplast. And those are the little green discs that are flattened that we've talked about in numerous chapters. And the cristae membrane would be the inner membrane of the mitochondria that we've also talked about in many chapters. But the purpose of these protein electron carriers is to kind of pass the electrons like hot potatoes down the series of proteins in order to gradually release energy that we're going to then use to make ATP. Okay, and so in cell respiration, for instance, NADH, which is a full taxi that we've talked about, is going to bring its passengers, which happen to be H+ ions and electrons. And this full taxi is going to drop its passengers off at the first protein, and the electrons are going to basically reduce this protein. Remember, reduction is gaining electrons, and so this protein is gaining electrons, so it's going to be reduced. And as soon as that protein's reduced, it's going to basically release a little bit of energy off of those electrons, and then it'll be oxidized while it transfers the electrons to this protein. And then this protein is reduced, and then it's oxidized and reduced and oxidized and reduced and oxidized and reduced, oxidized, reduction, oxidation, reduction, oxidation, reduction, oxidation, and reduction. The very last protein obviously will have to be oxidized as well, and there's always a final electron acceptor down here. In cell respiration, the final electron acceptor is O2, which is one of the reasons, or is the reason, that you require oxygen to live. It's not that the oxygen really gives you anything substantial in order to survive. The substantial part about oxygen is that it's an electron acceptor. It's the final electron acceptor. And so it sits down here at the end of your electron transport chain and accepts all of these low energy electrons, and they're low energy electrons because they all the way along this electron transport chain gave off energy gradually at each step. And remember, the importance of releasing energy gradually is that it's much more controlled, and it's a lot easier on the cell if it gradually releases instead of just releasing it all at once. Because remember, the previous slide is that if you uncontrollably release all the energy at once, you could have a cellular explosion of catastrophic proportions. Okay, but we don't release all the energy at once. We release it controllably. It's very important that you control the release of energy, and all of this energy right here is going to be used to actively transport hydrogen ions back to the high concentration side. And in class we're going to be going through examples of what it really means to be low or highly concentrated with H+ ions. But this would be the high side, and this would be the low side. And H+ ions are naturally going to flow through the membrane, through the ATP synthase enzyme, from an area of high to low concentration. But just like any diffusion, it will eventually reach equilibrium if you're not actively transporting these H+ ions back to the high side. And so we use this energy that is released from the electrons gradually during the electron transport chain to release or to transport actively these H+ ions from the low to high side. Remember, transporting something from low to high requires energy, and the energy in this case is going to come from the electrons as they gradually release that energy. Okay, so let's talk about chemiosmosis in particular. Chemiosmosis requires a membrane protein that is also an enzyme called ATP synthase. ATP synthase, again, is an enzyme, and you know it's an enzyme because what do enzymes end in? Ase. And so some membrane proteins are enzymes, but not all membrane proteins are enzymes. Okay, so that's a good distinction. And so this enzyme is ATP synthase. It is a membrane protein, and it spans the entire membrane. It is in the cristae membrane of the mitochondria, and its whole job is to synthesize ATP, which is why it's called ATP synthase. The root word of ATP synthase is basically synthesize or synthesis, and ATP obviously is ATP. So ATP synthase is an enzyme that is in charge of synthesizing ATP. And how does it do that? It's going to use a flow of hydrogen ions, which are also protons. They're going to flow through that ATP synthase down a concentration gradient, which means they're going to flow high to low. And in order to get from the high concentration side to the low concentration side, they have to flow through that enzyme, and it acts just like a channel protein that we've talked about in Chapter 7. These hydrogen ions are, like I said in the previous slide, actively pumped back to the high side to constantly keep that concentration gradient stable. Okay, I mean, you've got to maintain the concentration gradient. If you don't maintain a concentration gradient, your ATP production will cease, and if the concentration gradient or ATP production dries up or quits, then the cell's in TREBS. It's in big TREBS because the cell needs a constant supply of ATP to do all of its cellular games. Okay, so here's a picture to show you chemiosmosis. Notice that the high side is right here, high H+ ion concentration, low H+ ion concentration, and this would be, if we were talking about the mitochondria, this would be the inner mitochondrial space. So intermembrane space. It's the space between the inner and outer membranes. Down here on the inside of this membrane would be the cristae. Okay, cristae. So we have the inner mitochondrial space, or the inner membrane space. We have the cristae, and the high concentration ions, or the high concentration of H+ ions, are going to flow through the ATP synthase enzyme and are going to turn a turbine, so to speak, down here in this bigger bulbous area of this protein complex. And as these H+ ions flow through this ATP synthase enzyme, they're going to turn and power this ATP synthase wheel, which is going to phosphorylate or add this phosphate to our ADP molecule, which is going to ultimately produce ATP. And it's going to constantly produce ATPs. In fact, every 3 H+ ions are going to produce 1 ATP. So you can tell how many H+ ions there are and how many ultimately H+ ions it takes to make ATP. 3 H+ ions is what it takes to make 1 ATP. This is the electron transport chain, and remember, the electron transport chain is the specific structure that is going to actively pump these H+ ions back to the high side from low side. This is active transport. It's an active transport process, and it requires a membrane protein, which are here in the electron transport chain, and it requires the solutes, in this case H+ ions, moving against a concentration which is low to high and requires energy. The electrons, like I said, are going to travel down the electron transport chain, and they are going to release some energy gradually over time, and all of the energy that is released as those electrons move down the electron transport chain is going to be used to pump these H+ ions back to the high side. Okay, so run through it one more time. H plus ions are going to move through ATP synthase channel protein from an area of high concentration to low that doesn't require energy. It is a passive transport process. As the H plus ions, in fact 3 of them, move through an ATP synthase, there's enough energy at that point to power this turbine, which is going to add a phosphate to the ADP molecule. That's called phosphorylation. And once the ADP and inorganic phosphate join, it produces an ATP. Remember, ATP is adenosine triphosphate. Adenosine diphosphate is ADP. So ADP has 2 phosphates. ATP has 3 phosphates. How do you get 3 phosphates from 2 and 1? You smash them together. Okay. So that's chemiosmosis. Chemiosmosis is the main way to make ATP. I say here that it's one way to make AT, but it is the primary way in which we're going to make energy. And the majority of the energy that is made in a cell is made this way. This is the most important way to make energy, ATP. Okay, and so what other way, or what is the other way that we make ATP? And the answer is substrate-level phosphorylation. Substrate-level phosphorylation is the other way to make ATP, and it doesn't require a membrane like chemiosmosis does. It doesn't require ATP synthase, which is an enzyme like chemiosmosis does. It does, however, still require an enzyme, and this purple object here is our enzyme. And so it's still going to be an enzyme-mediated reaction, meaning it's still going to require an enzyme, but it's much simpler than chemiosmosis. Chemiosmosis is a fairly complex, abstract process where you have moving ions that are flowing through a channel protein and have to be actively pumped to maintain a concentration gradient. But this process is much simpler. It doesn't require membrane. This can happen anywhere within the cytoplasm or anywhere within the matrix, and all you need is an enzyme and the proper substrates. The substrates happen to be any organic molecule that has a phosphate attached and ADP. And all this enzyme is going to do is it's going to take in this organic molecule, but again, it has to have a phosphate. And this enzyme is going to basically take the phosphate from one molecule and it's going to bond it to our ADP. And when you take a phosphate from a molecule and bond it to ADP, you're going to come up with ATP and a molecule that now is phosphateless. Notice the molecule shape changed, and that just is structure dictates function. And so if the function and structure of this molecule is such that it has, it's structured this way because it has a phosphate, then obviously it's going to change slightly if we take the phosphate from it. And so again, the only thing you need to know about substrate level phosphorylation is that it requires an enzyme. It's a much simpler way to make ATP. It takes an organic molecule with a phosphate, takes the phosphate from it, adds it to ADP, and produces ATP. It produces much less ATP though. It's a much simpler process. It happens anywhere the cell needs additional phosphate or additional ATP. But it doesn't produce near the quantities of ATP as chemiosmosis. Chemiosmosis is the first choice of ATP production. Substrate level phosphorylation is the second choice. Both of which are important, both of which are needed, but you could think of substrate level phosphorylation as a way of making bonus ATP, and chemiosmosis is the way of making the normal amounts or the normal quantities the cell needs. Any way of making extra ATP is still good and still welcome within the cell. So I think that's where we're going to end it. If you have questions, make sure you bring those to class. See ya.